Well, the last PPT was regarding oxytrophic mutant. This PPT will be regarding overproduction of primary metabolites, resistant mutants. So we are just going through a lots of examples. You need not write all the examples in the exam. This is just for you to understand the concept. You can write any one example. So before we go to resistant mutant, one more example I need to quote. We can quote for oxytrophic mutant. In this example, you can see that glutamate is getting converted to arginine. Arginine is the end product in the pathway. But in this pathway, you can see there is one intermediate called ornithine. This ornithine is having lots of commercial application worldwide, and people want this ornithine to be commercially produced by microorganism. But one problem here is ornithine is the intermediate. It is not the end. It is not the end product. It is intermediate. So what we have to do, just you can know. By now you will be very clear that if you want ornithine to be produced in large scale, the only thing you have to do is you have to mutate the enzyme that converts ornithine to citrulline. By doing that, what will happen is you will be isolating oxytrophic mutant. Oxytrophic for what? Oxytrophic for arginine. That means the organism cannot produce arginine, because arginine, as you see in the diagram, arginine goes and controls the enzyme that converts acetyl glutamide to acetyl glutamyl phosphate. So because arginine is an end product and it does feedback control system, you are blocking the enzyme that converts ornithine to citrulline. So ornithine will be overproduced, arginine will not at all be produced. We call them as oxytrophic mutant, and you can give arginine, very less arginine in medium, the organism will grow. So again, this is one more example in the industry of how um, knowledge of the metabolic pathway is used for overproduction of metabolites in the industry. Now coming to our resistant mutants, as I have already told you, the difference between oxytrophic and resistant mutants do not recognize the presence of inhibitor. It means here end product will be produced, but the end product cannot bind to the enzyme. So such type of mutants are called resistant mutants. Let us see few examples. So here you can see here A is converted to B, C, D and E. In oxytrophic mutant, whatever we studied till now, we were interested in intermediates. We were interested in C, overproduction. But now what you are seeing is we are interested in end product E itself. But E, whenever it is produced, you know it will go and control the first enzyme. But you want E to be overproduced. This is the circumstance during which we will isolate resistant mutants. So the concept what people follow in the industry is analog resistant mutants. You see, we want E to be produced, but we don't want E to go and do feedback control. What will E basically do is whenever it is produced in excess, it will go and bind to enzyme E1, but we don't want the end product to go and bind to enzyme E1. For this, in the industry, they follow a technique called as analog resistant mutant. So resistant mutants is called as analog resistant mutant. What it is, we will see with few hypothetical examples. Okay, or with live examples we will just see. Here we want to produce lysine in excess. Lysine is a very important amino acid. After glutamic acid, lysine is having huge market value. We want to produce lysine in excess. This is the example which we have previously quoted also, but we are going by a different way here. Earlier we studied it in terms of oxytrophic mutant. Here we will study it in terms of resistant mutant. Example here is Brevibacterium flavum. This bacteria is known to produce lysine, we overproduce lysine by a process called analog resistant mutant. Analog here means analog resistant means here we are using a chemical which is analog to lysine. Analog to lysine means looks like lysine, structurally looks like lysine, but functionally it is not a lysine. Such compounds are called as analogs. So we are using something here which is analog to lysine. Example is S amino ethyl cysteine, AEC we call it as. See in the diagram, just observe, just I wanted to make things clear to you. What is lysine doing here is, whenever lysine is produced in excess, what it will do, it will go and bind to enzyme. which converts aspartate to aspartate semialdehyde. You can see the arrow mark. We want the lysine, we don't want the lysine to go and bind. That means what does it imply to you is we want the enzyme not to recognize lysine. That means for this we use a concept called as analog. Analog means looks like lysine but it is not lysine. That concept is used here. Students, one thing you have to understand, we follow a technique called as gradient plate method when it comes to isolation of analog resistant mutants in the laboratory condition. Just listen to me carefully. The concept here is you can just see a Petri plate there. It two types of media we are preparing in the Petri plate. You just observe it. Arrow marks is written. First you take a sterile Petri plate, you pour medium. You pour the medium slantingly, or you keep the plate slantingly and pour the medium, so that media will get solidified in a slanting way. The first medium what you are pouring is analog supplemented medium. That is what you are pouring first. After the media gets solidified, you remove the wooden block, you keep the plate straight. Now you can see the media is prepared slantingly. Now after keeping the plate straight, now you pour one more medium, normal medium. So two types of media is prepared in a single plate. First you are pouring first what you are doing is you are keeping the plate slantingly and you are pouring the medium containing analog. Once it gets solidified, you remove the wooden block, keep the plate straight, then you are pouring the second medium which doesn't contain analog. So basically you are preparing a Petri plate containing two medium. The bottom medium containing analog, the upper medium does not contain analog. What is, again I told you, analog, why I am telling analog is you will, yeah, okay, you will, this example you will maybe you may understand it properly. Once this media is prepared, what we do is we will take the organism and we will do mutation as usual. We will expose it to UV light. After exposing to UV light, we have to plate it on the surface of this medium. So when you plate it on the surface, you can just see why we call it as gradient plate. This plate is called gradient. Why? Because you can see one corner of the Petri plate, the analog supplement will reaches the top, and the opposite side of the Petri plate, the analog medium tapers down. That means why we call it as gradient plate, because where analog medium, the bottom medium containing analog, where it reaches the top, their analog concentration is very high, and the other side of the Petri plate where analog medium tapers, their analog concentration is least. So in single Petri plate, the concentration of analog in one corner will be very high, in the other corner it will be very less, in the middle it will be reducing or increasing. So we call it as gradient plate. Gradient means the concentration of analog from one end to another end constantly increases or decreases. Now when you do the plating, what is analog? Analog is called toxin. See, for example, lysine. What is lysine? Lysine is the end product. And what is AEC? Amino ethyl cysteine. That is called analog. That means I told you analog and AEC and lysine both are analog to each other. That means AEC will mimic lysine and it will go and bind to enzyme. So wherever lysine has to go and bind to feedback control system, that feedback will be done by AEC. So when you put this AEC in the medium, analog of lysine, what will happen? Before lysine gets produced, AEC will go and bind to the enzyme. Because of this, what happens? AEC can also be called as toxin, highly toxic compound to microorganism. The microorganism will not grow at all when you give AEC in the medium, because AEC structurally resembles lysine. Instead of lysine, it will go and bind to enzyme and it will stop the enzyme from functioning. It will do feedback control of enzyme which is done by AEC. Because AEC does feedback control instead of lysine, we call AEC as a very toxic compound. When AEC is added, organism will never grow, they will die immediately. We call them as toxins. So as I told you, you are exposing the organism to UV light, and when you plate it on gradient plate, you have to spread plate on gradient plate. You can just see in the below diagram that the organism will grow luxuriantly in one corner of the petri plate. You can see the black color zone, that is the organism growing very nicely in one region, because the concentration of AEC is very less. That is why we call it as gradient plate. Wherever AEC concentration is very less, the organism is growing luxuriantly in the form of a mat, and wherever AEC concentration increases, the organism will not grow. That is why half of the plate will have nice growth, the other half will not have growth, because AEC concentration is more. If you see one or two colonies growing in AEC concentration, high concentration, those organisms are called resistant mutants. Resistant here means they are resistant to AEC. So naturally, if they are resistant to AEC, what does it mean? They are resistant even to lysine also. That means those organisms that grow in high concentration of toxin called AEC toxin, they are resistant to AEC. Means AEC cannot go and bind to the enzyme, no feedback control is happening. Such organisms will be naturally resistant even to lysine also. That means lysine also, because lysine and AEC are analog to each other. If they are resistant to AEC, they are resistant to feedback control by lysine also. So such mutants are called as resistant mutants, and this is how we use the technique to isolate resistant mutants, and this is how we produce lysine in excess. I hope you have understood the concept. You just can just go through it. You will be clear with the concept. With this, we are completing overproduction of primary metabolites. As I told you, this is a huge topic. The first unit is very important. Exam point of view, strain improvement technique has been asked every year for 15 marks. So lots of importance we will be giving to this particular topic as such, because it is a constant question for 15 marks. So with this, we are in a halfway stage. We have completed overproduction of primary metabolite. From now onwards, this is most important what you have studied till now. From now onwards, we can move little faster. 2 or 3 more session, we may be completing this strain improvement topic as such. So next topic for us is overproduction of secondary metabolite. Primary metabolite you have studied. Secondary metabolite we will see in detail from the next PPT. Just an introduction for secondary metabolite. You know the difference. Secondary metabolite means what metabolites produced during stationary phase of the microorganism, that is during the stationary phase means example antibiotics. Whenever microorganism faces tough condition, unfavorable condition, whatever product they produce during unfavorable condition is called as secondary metabolites. So that we will see this is also very important in industries produce primary metabolite, but there are industries which produce secondary metabolite also. As I told you, antibiotics, huge market value is there for such antibiotics. And here also blueprint of the organism is essential. It doesn't mean that it because it is antibiotic, without knowledge of microbial blueprint or the metabolic pathway, you can't do any strain improvement. But people without knowledge only all these days they have been doing. Penicillin, streptomycin was produced as early as 1940 to 1960. People were producing them in large scale. During those days, they didn't have knowledge about the blueprint of the organism nor the metabolic pathway control system. But still, during those days also, strain improvement was being done. They were producing penicillin, streptomycin in very large scale during those days. The only difference is that during those days, without knowledge they were doing. Now with knowledge we do strain improvement. Without knowledge, what they used to do during those days was random mutagenesis, because they didn't know the pathways clearly. how penicillin is produced, how streptomycin is produced. They were just doing mutation randomly, so we call it as random mutagenesis. During those days, what they used to do was brute force. Brute force means simply they will be doing mutation, they will be plating. So it was something skillful process they were doing, very laborious work they were doing, and they were following a technique called as empirical method. This what is this empirical method, we will see it in the next slide. So we are starting with overproduction of secondary metabolite in our next class.